Hello, my name is Asija Rice. I attend Southern University. I'm an urban forestry major, and I'm a senior from New Orleans, Louisiana. This summer, I worked with developing pollen sampling for methodology for neon condensite for a pathway program for LSU this summer. Okay, I worked in Hammond Research Station with Dr. Yan Chan. His research station is where they do different trials and different evaluations for different super plants around, the United States. Definitely show like what type of environments they grow in and what type of soils and what type of bugs we can infest them with. Okay. My objective for this project is to find a methodology to collect pollen from a crepe myrtle and also collect the blooming and flowering time for this crepe myrtle. And you'll see that I'll try to collect the best quantity, the best quality of the pollen. Okay, the crepe myrtle, it has over 400 varieties. It's a summer blooming shrub, a tree. You see them everywhere. I never knew that crepe myrtles was everywhere, literally. They bring in $66 million a year. One thing about this flower, it's constantly blooming for 3 months straight. Wash off, bloom again, wash off, bloom again. Also, it's going through this process that has been infected with the crepe myrtle bark scale, which is really bringing in damages since 2005, and where I'll go on to the next slide. And you'll see that the crepe myrtle will be damaged. It'll turn the bark black. It'll look like it's mold. It messes with the production. The tree will not bloom if it's younger. And then if it's get affected for a long, various time, like it's a 6-foot tree, you'll be wondering, okay, I got this crepe myrtle in my yard. It haven't bloomed for 3 months period. It may have been affected by the crepe myrtle bark scale. You can see on this map that this crepe myrtle has been everywhere around Louisiana, and from what I know, it has spread it. Okay, this is the most important part of my project. I studied the phenology part of the phenology part of crepe myrtles, and also I had to come up with different methods to collect pollen from a flower that was this small, and it was just me. And but basically with the phenology part, I had to basically learn everything about this tree. When it bloomed, how long it took to bloom, what time did it spend, what time did it bloom, from like hours if I sat outside and watched it bloom from 5 o'clock to 12. Just different bees, different walls that visit this tree. Okay, let me start off my methodology part. My first trial was horrible. Like, this is my test trial. I made 20 tubes out of tubing, the little needle part, and cigarette, the filters, you could say. I had a vacuum this size. The original plan was to get 2 ladders from the stand on the ladder, put the vacuum on one side, and suck pollen from this tree. Yeah. So what I had to change my whole plans. I took 2 Petri dishes. I put 4 buds in it. 1 in the AC, 1 was room temperature, to see when the pollen come out. I had to literally watch and see, okay, it took this pollen an hour to just come out in full form. So once I understood that the pollen was going to come out at a certain time, that I couldn't rush it, I also had to basically put it under a microscope, look, and see. I could basically see the pollen releasing just at a time. It may not look like it's releasing with an eye view, but it's a ton of it. So my next trial, I picked 6 Natchez white trees that are big, they're covered with white flowers. You'll see a lot of them on the ground because they're so delicate. But with this, I collected a cluster. Each cluster had 50 flowers on the cluster. I picked each flower off the cluster. I put them in. Each tree had 3 different containers. The sandwiches container, each container had 50 flowers in it. My first trial, my crazy self, took all the petals off, took just dissected the whole flower because I didn't want the trash in a vacuum. That didn't work. It didn't matter. On the next slide, you're gonna see why it didn't matter. Everything molded. You could tell the different environments. You could tell that the pollen released differently from when it was just outside, and then when I brought it inside, it was just two different the outcomes. So my next replication, I collected 900 flowers. This time I didn't pull them apart. I just put them in a container. I shook them really hard. I took the sponge paintbrushes, the sponge paintbrush. I wiped it around the container. I took the vacuum and I sucked the pollen off the vacuum. So far, I collected two. It was like 2 tubes. They wasn't filled at all. I came across the pollen was sticking to the cotton. So even if I did have a collection of the pollen, it was all on the cotton. I had to throw it away. So I wrapped tape around the cotton, put it back in there. So it was just, I can say from the experience, I can tell y'all what not to do. Okay, you can see like what I was talking about for my experiment, molded on the vacuum, 2 or 3 at the most for different. The reason I'm gonna say it probably molded was the air temperature. I had them in a container. Flowers don't bloom in a container. I know not to do that again. I'll also be sharing this with the LSU team that's gonna actually be doing this experiment. So they're like, okay, we're not gonna collect the pollen and put them in containers. Each tube was weighted before and weighed after. I know it's not much difference, but this was the weight when I put the pollen in the tube. Okay, this is flowering phenology, my favorite part. For 10 weeks straight, I went outside and I collected the bloom week for Crepe Myrtle, a different series. Also, for 6 weeks straight, I collected the new bloom, spent bloom, and full bloom on each for 10 different trees. So actually I went out, if it's a tree right here, I counted 1, 2, 3, 4, 5, every other week. And with the blooming times, I collected the hours of the day. Like I said, I'll sit outside from 6 to 12, 12 to 1, depending on, I collected data when the pollinators came to visit, who was visiting. I didn't know that flies collected pollen. So yeah, it was different. This right here is showing the Crepe Myrtle blooming. And over the time, the Crepe Myrtle only lasts for 1 day. If it rains, it's not going to even last for that 1 day. So as y'all saw, those begin to spent. This will be a new bloom, and this will be a full bloom. A full bloom will be, if it was a whole video, picture of the cluster, all the flower be bloom. For instance, they'll look like this. These are mature flower trees right now. They're not to this full form. So most of the data I collected, I'm not gonna say it's accurate because they're still little trees. Right now, out of all the trees, I had 169 trees I collected data from. I narrowed it down to 29, and I narrowed them down by how popular they are. So we have the Black Diamond series and the Delta series, and also we have a farm series. It's Red Rooster, Crimson Red. And one thing about it, I know why they're so popular, they give off bright colors. Once you walk outside, you see them like, oh yeah, this is a Crepe Myrtle. Like. So, okay, this is a chart basically saying all the blooming weeks I covered for the Crepe Myrtle. And this is when I begin to pick out, this is my early bloomers and my regular bloomers. And I didn't really get any data like I did for the rest of them because they bloom later at the end of the internship. So to let y'all know what's going on before you have any questions, This is my weather chart, basically saying the amount and average percentage of rain over the 3 months. That'll be able to show y'all why my spent data was so high when it shouldn't have been. Okay, right now is my full bloom. From what I can tell you, are the crimson red and the plum magic will be your best cultivars to pick to collect pollen from when they do the study. So so you'll know that week 25 and week 26 of the year that plum magic and crimson red, for example, will be your main 2 cultivars that you'll pick to collect the pollen from. In the spent, you'll understand that over the first 6 weeks, that it was less spent, then more rain, and the higher the spent count. Okay, I'm trying to move faster because I know that I have a lot of charts.
I'll take your time, but don't.
Okay, right here is the total bloom. So with the total bloom, this is my favorite chart because it shows that when a team that LSU brings out to collect the pollen and collect the pollinators times, they'll be able to see that we're going to monitor plum magic, and we're going to monitor Delta Moon series, and that'll be our main focus instead of collecting from all the different trees that takes all the way to July to bloom. So if they needed to collect the pollen within that month, these will be the series that they'll collect them from. And they're like I said, they're long lasting, they're constantly blooming every day. And oh, my video's showing sideways. It was just a video of my 169 cultivars. At the end of the project, I would like to share that the methodology that I developed will be used for the LSU upcoming project. And basically, like I said, it will show them what not to do and when the best time for them to go out and see. So they don't have to guess and say, oh, we're going to go collect data from this Crepe Myrtle at 2 o'clock in the evening, but it didn't have a rainstorm at 12 o'clock. There's no pollen for you to collect. Also, I had to battle with different pollinators. I'll tie ribbons around the trees, and basically I'm gonna come back and collect pollen from you. When I come back, the whole blossom is just gone. It's not there at all. This project has given me the opportunity to switch my whole mindset. I never thought about going to school to get a master's. I'm open up to a master's degree now. I'm an urban forestry major. I want to reach out to communities, but this also has given me the opportunity to say, hey, this is the science part of it. This what can a tree do for you, and what can you do for a tree? Also, just to beautify your landscape. So this project here it tested my patience. It tested my analytical skills that I really thought I didn't have as much because I never did a research project before. And it was a real eye-opener. Like, I applaud anybody who has to sit down and watch anything become what it is. But the whole point of this project basically is the team is going to test the pollen the way it is before they add the neon condensate to it. Neon condensate that's in the plant may be dangerous to the pollinators. So right now we'll get the samples of the pollen, and then when they go back and spray the trees that's infected by the Crepe Myrtle bark scale with the neon condensate, we'll see if it's infecting the bees, the wasps, the flies. And as we know, it's already declined in bees. We don't need to spray the trees with more damaging projects to them, and it'll just wipe out. So this is my research work.